Humanity transitioned from hunting and gathering to fully developed agriculture and cattle breeding approximately 10,000 years ago. Today we are entering a new phase of human agricultural activity. It's a new stage of development when agriculture is becoming intensive. To put it plainly, farmers can no longer continuously extend the lands they cultivate. However, since the demand for agricultural products is growing, farmers need to think about means of increasing their crop volumes from the lands they already possess. To achieve this, crop production can get significant help from the magic of science called genetic engineering. It's a relatively young scientific direction and therefore still raises a lot of questions. In Kazakhstan, the place where genetically modified plants are being developed is the National Center for Biotechnology. One of the projects is the cultivation of genetically modified potatoes to produce high-quality starch.
В результате реализации данного проекта мы получили.
As a result of this project, we obtained potato tubers without the gene that encodes amylose. That is, from these tubers, we can already obtain starch containing only amylopectin. Amylopectin starch is in high demand in production.
В производстве хороший спрос. Амилопектиновый крахмал используется в пищевой промышленности, поскольку обладает
It is used in the food industry because it has a pronounced viscosity behavior and stability. It doesn't have an odor and can withstand temperature fluctuations. For example, amylopectin starch is perfect for ketchup production because it serves as a good stabilizer.
Используется для производства, например, кетчупа.
Genetic modification technology has been used for a long time. A great example is when a farmer selects the seeds of the sweetest fruit, grows them and repeats the process again. This is also seen as a focused improvement of specific traits. However, now there is no time for such a long selection process outside the laboratory, and the result is not always predictable. Working with genes gives more guarantees as it involves alteration of specific parts of DNA. Scientists do not just break and change something randomly. For example, to add sweetness to a watermelon, you need to clearly understand what parts of its DNA are responsible for the sweet flavor. It ensures that we can safely cut the DNA exactly where we need to modify something using the so-called molecular scissors. Furthermore, the cost of DNA reading techniques is declining, making it possible to read the genomes of organisms that have previously been experimented on and compare the changes.
Про CRISPR-Cas технологию, данная технология.
CRISPR technology allows to edit the RNA by introducing mutations into the plant genome systematically. Thus, for example, it is possible to improve drought resistance, productivity, resistance to biotic or abiotic factors, and other technological qualities without introducing a foreign gene.
Или же там такие технологические качества.
Of course, it's possible to imagine that a malicious genome change may theoretically result in the development of a toxic plant. However, in practice, GMO serves the public good. Harmful GMOs will not be able to pass assessments and end up on store shelves. International law requires every product to undergo rigorous testing to ensure it is safe for humans, animals, and the environment. Despite all the difficulties with development and safety testing, scientists are confident that in the future mankind will not live without transgenic plants and products. Genetic engineering can prevent famine or massive crop failures and minimize environmental damage. GMO crops can be watered less frequently and cultivated using no-till farming. This will not only save water but also reduce the impact of the greenhouse effect by decreasing the heat radiation from arable land. In addition, reducing the amount of agricultural equipment used in the fields will also aid in decreasing atmospheric carbon dioxide emissions.